Today, I'm going to talk about the need to think again and relearn research methods when working with farmer research networks, or FRNs. My ideas come from working with the Collaborative Crop Research Program over more than 10 years, and also from other programs that bring farmers and researchers together that I've worked with. And here's the basic problem. FRNs do research. It's in the name. FRNs involve networks of people collaborating together, farmers, extension agents, NGO officers, and others, and researchers. And researchers understand research methods. It's part of their training, their skill, their professional qualifications, and hence FRNs shouldn't have any problems with research methods. However, when we talk to researchers about the research done by and in FRNs, we often hear complaints, comments, criticisms such as, there's too much variation. How do we know about the validity of this research? Can it be replicated? Farmers' data is unreliable. We don't know what farmers are actually doing on their plots, and so how can we trust the data? And ultimately, can this be published? Students say things like, my supervisor told me to do it differently. So what's going on here, and how can we resolve this apparent conflict between what researchers believe is correct, valid, useful research, and the way that FRNs operate? That's what I'm going to examine in this talk today. I said research methods, and if we look at research methods, they're enormously broad. There's no limit. It's very hard to even categorize research methods and say, list them out in some way. But I've just highlighted, chosen here one way of trying to distinguish some methods which are used in our sort of business. The top level titles there, primary data, secondary data, synthesis, they're describing the sort of data that we're using. Primary data meaning data that you're collecting specifically for this piece of research. Secondary data means using data that already exists, somebody else's data that's been collected probably for another purpose for your research. And then synthesis means not necessarily using data at all, but taking ideas and trying to assemble them into some sort of more complete picture. And under each of those headings, there are all sorts of methods we can use. So if we start on the right-hand side, the synthesis, you've seen, I'm sure, examples of various models being used where we put together some description and try and figure out what is the model predicting about how the world works. We try and assemble lots of facts and make a comprehensive story about it, what I call qualitative synthesis. We can use what I tried to summarize as logic and deep thought. I've asked many groups, particularly students in East Africa, so tell us a great scientist. Who do you think was one of the best scientists? And lots of people mention Einstein, which is fine. He was indeed a very eminent scientist. And interestingly, he didn't use data at all. He never collected data. What he did was thought very deeply and logically and carefully about a problem and deduced something from that thinking. So that's the synthesis. Secondary data, we can do analyses based on official statistics. Several of you circulated recently a fascinating graph drawn by Pablo Titanel and just published about the importance of smallholder production in global food systems, for example. And it's all based on unofficial statistics. He didn't go and measure any farms to do that. That's an example of a meta-analysis where you pull together results that other people have put together and try to make some collective sense out of them. You've all seen lots of things based on remote sense data and other approaches which are sometimes labeled big data. It means using data that's being collected for other purposes, particularly when there's a lot of it, and trying to interpret for your own purpose. And then we have those methods which are about primary data, where you collect things specially. And it's helpful to divide those into intervention-based methods and observational methods. Interventions are where you are deliberately changing something to try and see what happens. And observational, where you're trying to observe what's going on without necessarily interfering or changing it. And so those observational ones are surveys, and we do surveys of people and households and farms, and we do surveys of ecosystems and environments, surveys of soils and surveys of trees and so on. We do surveys of markets to see what's going on in markets. We do less structured and formal observational studies of things like focus group discussions and collecting data from key informants and that whole array of PRA tools that you're familiar with. Recently in East and Southern Africa, we've been using these participant observation methods to try and understand more about how FRNs are working, actually. And then on the intervention side, we've got experiments, lab experiments and field experiments, and experiments that take place at bigger scales, things which in development literature are often called RCT interventions or action research models and so on. And for all of these, there's any number of details and variations on the methods. Almost all of them you can think of doing your research in a qualitative or quantitative way. There are overlaps between them and ways in which they're integrated and put together. There are principles and bits of theory that support most of these. There are new ideas coming up all the time. And pretty much any of these research methods could be useful to FRNs, and quite a few of them I can think of how FRNs or groups that work like FRNs have actually used these things already. Okay. So there I'm just stating the case. Research methods are huge and broad. For this discussion, I'm going to concentrate on just one little bit of this great big set of methods, and that's the field experiments. And the reason for that is that although all these methods and ways of doing research are available and are used, from what I understand of the way FRNs work, nearly all the research done by nearly all the FRNs is based around field experiments. So for this talk, I'm going to concentrate on field experiments, but the key messages I have are going to be relevant to almost any of the research that an FRN might engage in. All right, so let's think about experiments. And these field experiments done with small plots are such a standard and well-recognized, well-developed part of agricultural research that everybody learns about them. Anybody learning agriculture or some specialization in agriculture like soil science or crop breeding or learns about experiments. And the things they learn, the sort of archetypal experiment looks something like this picture I've got here. This was, I could tell you how we took that picture. It's an aerial photograph of a small plot experiment, and it was taken long before we had drones and digital technology. So you might like to wonder how I managed to take. I took that myself. So you might like to wonder how I did that. But there we are. It's a field experiment taken vertically, looking downwards, and you can see on the screen there I've listed some of the key principles, if you like, or topics around which there are principles to do with these experiments and how we set them up. We carefully choose the sites we use and we characterize them. I've actually pointed this picture includes in the very top in the very top left, you can just see from the air, a soil pit was dug so that they could understand exactly what the soils looked like down to 2 meters. We have these well-defined treatments that we're comparing. We have random allocation of treatments to plots. We have replication. You can tell here, even without knowing what these treatments are, you can see that each one seems to come several times. We aim to control variation. You can see everything looks beautifully homogeneous. This crop, everything is even, the soil looks even, the crop's been very evenly planted and weeded and everything. That's because we're aiming at precision. We'd like to get precise answers. And part of that, and then when we're collecting measurements, we're measuring these things, we make sure we use objective measurements. We weigh things and take scales to them and use rulers and moisture meters and things like that to collect the data in these experiments. And that sort of research, it was first systematically described in 1925. Fisher wrote this famous book, research method. What's it called? Research designs for research workers or something like that. And there is no question that it's been extremely effective. A large part of what we know about agriculture and all its many types and sources is based on this research using these designs. So I don't want to say this is wrong or this is useless. It's not. It's been extremely effective, and for the last 100 years, it's given us this very superb understanding of how agriculture works, the science of agriculture. For that reason, these are methods that are included in every master's training agriculture, and maybe in bachelor level training. Most master's students doing projects have quite strict requirements about the sort of experiments they do and what qualifies as a valid experiment for their project. And that then means that researchers and institutional organizational views as to what good and valid research is about is based on the same sort of thing. Right? And this is for field experiments, but I could start listing the similar sets of principles that have been derived about almost any of those other research methods that I listed up there as well. Okay? So we have the science, the theory is well established. You can find it in textbooks and so on. There shouldn't be anything controversial about it. During the what I'd call the participatory revolution in the 1980s and 1990s, when agricultural researchers, particularly in the sort of agronomy area, started saying, we need to be participatory and we need to do things with farmers and we need to do them on farms. Then the same sort of thing was done. Okay, these design principles were adapted for doing in farmers' fields. And I've just this is a made up example, but the same 4 treatments which were in my previous slide, I then copied and pasted and showed them scattered around here. And you often hear things like, I used a design where farmers are replicates. Right? And even very highly regarded proponents of participatory research principles describe this sort of thing. So you can find, for example, the textbooks by Jackie Ashby, who was a leader in participatory research and participatory research methods in the early 90s. She describes trials exactly like this. And when I see farmers are replicates, I think, wait a minute. What if somebody said that about me? What if somebody said, okay, these people are replicate. Me, Rick, I'm a replicate of something. I would feel quite offended and think, no, I'm not. I'm different from everybody else. And I have my own interests and values. And I want to feel that autonomy and responsibility things. I don't want to be thought of as a replicate of anything. And when we look closely at any group of farmers anywhere, of course they vary in everything that matters as much as any other group of people vary. They're not replicates of anything. So what's going wrong when we end up with designs where we say farmers are replicates, when clearly farmers are not replicates of anything? What's happening? Let's jump now to look at FRN principles. The principles that we've written down and try to follow when we're working with the FRNs fall under these three groups around who the farmers are, and about the research, and about the networking. And the first two, I've highlighted those that, in my opinion, should have a big impact on the way we do research. In other words, the research methods. Co-creating the research agenda. Okay, farmers engaged throughout the research process. Farmers' capacity to learn together. Okay? Think about what those could mean for research methods. Okay, Peter's already asked about how do we engage farmers in data analysis, for example. Well, this capacity. What do we do about farmers understanding those research design principles? How can they be engaged in the research process if some of those things that we believe are basic to doing good research are not things farmers understand? So we have to do something about that. The second one, research addresses farmers' problems and opportunities, continually adapted. Right? Co-developed research plans. Where does the co-development come in those sets of principles that I outlined on that nice, neat experiment that was there? It's based on sound and appropriate designs, but they're also participatory designs. What happens when farmers start participating in the designs? Are we going to end up with things that look like this if farmers have participated in designing them? We're going to involve relevant local, indigenous, and farmer knowledge. It's going to be integrated into the research. Where does local knowledge get integrated into here? So there seems to be possibly a bit of a contradiction between these principles that we've signed up to collectively and we try to follow, and those things that we believe we know about what makes good research. And to try and sort of unravel some of those contradictions, I'm going to work on a particular, I'm going to go through a particular example. Right? I should just point out here, I've put at the bottom of this slide, these principles, which are on the screen now, these FRN principles, are certainly not the principles that Fisher used when he wrote his extremely influential textbook on how to do research. And that's really the key to the unraveling this distinction. Right? Fisher was using principles, but they weren't these ones. All right. So what happens in practice? Well, a few years ago, prompted by a couple of FRNs in East and Southern Africa, we wrote a list. We took this capacity, farmers' understanding of research methods, particularly around experimentation. Let's take this principle of farmers increasing their capacity to do this kind of work. Let's take that seriously. And we started trying to outline, if you like, a sort of curriculum for what it is that if they're going to do useful, informative experiments, what do they need to understand? And we started writing down a list of new principles of experimental using experiments to learn things, and then some sort of explanations about them. And there are about 10 of these, and at least one at FRN did start using this and came back very positively, saying this is useful. We've started using this to discuss things with farmers and to help them understand and gain new understanding. So that was quite nice. But I realized after we put that together that it wasn't really enough. Okay? Because this is mainly aimed at sort of helping farmers to think the way that we are thinking. And it's not doing too much of helping researchers take a perspective that is close to farmers' perspectives. All right. So what else is there? Let's think of a particular example. And I can't see if we've got anybody from the Best Bets project in Malawi online at the moment. And something that the Best Bets project there has been working on for a long time. Farmers intercrop sometimes cereals and legume to benefit production and soil health. And they had this idea, what happens if we use 2 legumes rather than just 1 legume? What the idea that they call doubled up legumes. And so the FRNs have done a trial. They've actually done many trials on this, comparing single and doubled up legumes. And there have been many farmers groups involved and hundreds of farmers doing these trials. And amongst the findings that they've generated, they found that the results, that is the impact on almost anything you choose to measure, like production of the maize or measures of soil health and so on, depend on the soil type and what status it's in. They depend on things like weeding intensity and whether you do your planting at the right time. Particularly because of that weeding dependence on good weeding, on timely planting, they are strongly influenced by labor availability, and that there's a strong gender dimension to that. It turns out in some parts of Malawi there's also a strong gender dimension in relation to soil type and soil health as well. And so what we're seeing is, instead of that experimental design principle of control the variation would not allow you to discover these really important findings. Because the way the trial was done, involving lots of farmers that had different soil types, and some of them weeded early and some of them weeded late, and some of them planted early and some of them planted late, and some had plenty of labor available and some were really pushed for labor and so on. We, together with farmers, could interrogate the data and find out that these really rather important results. But if we'd followed that first design principle of control the variation, we wouldn't have been able to discover that at all. The standard way of doing these experiments is, let's make sure that everybody weeds on time and everybody weeds on the same way and everybody plants at the same time. Right? And we then have got no results about this. The concept that there is an effect of the treatment to be measured is flawed. And this actually is the core concept that Fisher and all his successors were using when they derived their principles of how to do experiments. They said, the aim is to measure the effect of the treatment. And in this case, there isn't an effect of the treatment. There are lots of different effects of treatments. So the whole basis for the way we learn has shifted from trying to measure the effect of a treatment and thinking about all this variation as sort of noise and we need to control it because it's hiding what we learn about the treatment, to saying all that variation between farms is now a source of information, it's not noise, and it's making it able for us to learn about these multiple effects of treatments that occur in different situations. Okay? So the key point here is that the basic concept that Fisher and his successors were using is not relevant to our problem. So we shouldn't be surprised if the design principles that he derived are not relevant to our problem. Let's look at another example from the same research area. In that doubled up legume trial, we could say, all right, we're going to have groundnut plus maize. That's what's going on at the moment. The doubled up idea is we have groundnut and pigeon pea as 2 legumes plus maize. That's a distinctly new one, and some sort of current practice or baseline. Now, you've all seen this example. Current practice varies between farms. What farmers do in their standard maize crop varies enormously. Whether they use fertilizer, how much fertilizer they use, manuring, variety they use, the spacing, and so on. And when we have a trial like this, and of course, sorry, I should say, farmers want to use current practice in the design because that's what they want to compare. They want to know, is it worth my while changing? Should I change from what I'm currently doing at the moment to something different? Okay, so I better include what I'm currently doing as one of the treatments. And researchers complain, we don't know what these treatments are compared to because current practice is different for every farmer. And this makes you think about this question. So what are the real objectives of this trial? If the objective is, number one, allow researchers to estimate the effect of doubled up legumes, is that it? Or is it more like number two, where there's an objective of helping farmers understand whether doubled up legumes will be useful for them? And what are the factors that make it more or less beneficial? That first, objective 2A, farmers need to compare things with their own current practice, and they'll make up their own minds whether this new idea of doubled up legumes is actually going to be beneficial for them. The second one, 2B, farmers learn through their networking. They hear experiences of other farmers. They might see it in data, or they might just hear it in terms of people talking about it, and begin to understand when the doubled up legumes really are effective and when they aren't. And if your objective is number one, okay, to estimate that effect of doubled up legumes, my conclusion is you need to do a different sort of trial. Why are you doing an FRN type trial if that's what you're trying to find out? One more part of this design problem. One of the FRN principles is that we will negotiate the design. Right? And the treatments compared are a pretty basic part of the design. So we have some negotiation about the design. And what happens when we negotiate? The group there in Malawi actually reported this and said, look, what we find are sort of different groups of treatment, different types of treatments. So those that indeed everyone wants to include. So the groundnut plus maize, which is the intercropping people know about at the moment, and then the doubled up legume as the innovation. Yep, everybody wants to try those. What about the maize alone? That's really only interesting to people who consider growing maize alone. Those that already only intercrop with groundnut, that's not really an interesting treatment to include. So only some groups, these are farmer groups, only some farmer groups included that one. Some farmer groups, or one farmer group I've represented here, said actually we're also interested in soya. So they put in soya as well as the groundnut plus maize treatment. And then you've got a few individuals that said, hey, I've learned about tephrosia as a legume. I want to try that. And others said, no, we don't do that. Well, so here's an individual who tried tephrosia, and here's another one that tried bambara nut, but it's not included. It's not done by everybody over the whole trial. Now this is based on what actually happened in that project, but this is not an accurate representation. I didn't have their own data. This is my sort of simplified version of what really happened. And when you get that sort of set of treatments, you hear these criticisms from researchers. The design is a mess. I mean, look at this. We've got unequal numbers of replicas. We've got things that appear in one place everywhere. We've got others that appear only in some places. We've got these odd ones that appear in one farm only. You won't find that sort of design described in any textbook. You can look through all these standard books on experimental design, and you will never see anything like that. It can't possibly be valid if it's not in any book. And anyway, how do you analyze it? Try and find the analysis of variance option in your statistics software to analyze that. You won't find it. Right? On the other hand, okay, the design, in some ways, it looks a mess, but it's actually very logical for its purpose. Okay, it's been negotiated, and there are these things that everybody's interested in, and these things which members of a particular group are interested in, and those things which some individuals are interested in. Why shouldn't we do it like that? Right? It is a logical response to the problem and our FRN principles. Right? It's not described in any book. That's true. But so what? No. Not everything in the world is described in books. And if it's not described in a book, it doesn't mean that it's not useful or valid. And the idea that it can't be analyzed, I'm afraid that's just untrue. Two things. One, analysis isn't the same as doing analysis of variance. But even if you do think of analysis in terms of analysis of variance, there are things that you can do with data from designs like this. All right. Okay, I'm ticking the last example from this trial, and this is to do with what we measure. So the things we like to assess, maybe yield or pests and diseases, or the labor involved, or the overall benefit from which we might think about things like adoptability or usefulness. The standard way of doing this would be yield. Well, we know how to do that. We go at harvest time, and we harvest, and we weigh it, and we maybe take samples for drying and so on. Pests and diseases, yep, there are standard methods for counting. We can use scores to measure the amount of damage. We can count the number of insects on plants of maize and so on. Labor, we can work out the number of days and hours it takes to do things and so on. And these overall benefits, you've seen these cost-benefit analyses done. You can calculate things, put in some prices and costs, and come up with some nice calculations. Now, if you though negotiate this with FRNs, you find a completely different set of ideas for how assessment might be done. So yield, most farmers don't need to go and measure yield in terms of kilograms per hectare. They can look at this, they can harvest it and think, this is great. This is much better than I usually get. I'm really impressed with this. Or think, I'm not very impressed with this. I'd have got pretty much the same anyway, or this is worse than what I usually get. So that there are options, and the two options I've shown here are not the only ones. For any of these things we measure, there are many options. And the point is that each of these options has some advantages. So in green here, yeah, these sort of standard scientific ones that researchers like are objective and unbiased, meaning that they're sort of repeatable. Somebody else could go and do it and come up with probably much the same answer. On the other hand, these ones which are sort of farmer-oriented, they're quick. Farmers are in control in the sense that they collect their own data. They're adaptable to any sort of problem. Red are some of the negatives involved. Okay, some of these measurements here, the standard ones, require technicians. You have to, go with a scale to the field, and you have to dry samples in an oven and weigh them again and do some calculations and things like that. And most importantly, they don't necessarily capture things that farmers are most interested in. On the other hand, these farmer ones, it's true, maybe they're not comparable. Maybe comparable, for example, if we do this in Malawi and we do it again in Kenya, then I'm not sure I'm measuring or assessing the same sorts of things. It's hard to aggregate them. In other words, put together data from different places and come to some overall conclusions, because there's a lot of things which are locally specific in each one. So the approach you use is not going to be to say, this is the way on the left here, this is what we know how to measure these trials. It's what agronomists have always done. We need to be saying which approach to measurement actually meets FRN and farmer objectives and is aligned with principles. And in practice, we're going to adapt and merge both rather than assume that one way is the right way. Okay, so trying to think about how we do these designs. If we take an objective, so I've just written this down, understand the impact of doubled up legumes on soil microbial diversity. This is a great topic for a student project, isn't it? Okay, hot topic, soil microbes. What do? We look at our experimental design principles. We've got other research principles. We think about context and logistics, and we can come up with a design. And I'm pretty sure that in this case, we'd come up with something that looks like a conventional experiment, rather like the one that I showed you at the beginning. If we have an objective like this, that FRN members, as the farmers and researchers together, want to understand, I gave a specific example here, maturity length of pigeon pea varieties and how we can manipulate those to change the performance of doubled up legumes, then the process is different. We're going to take FRN principles and we're going to take FRN design principles, and we're going to do some negotiation. We might do some capacity building, some learning of more about how things work, and we're going to come up with some adapted design, which is not going to look like the conventional design. So there's a different process to go through to end up with a design. I think when I wrote this down, I realized that we had been a bit negligent. There's some tasks which we really need to do. And these are, first of all, can we write? Are we now in a position to write down what some of these design principles for experiments done by FRNs? Can we write down some of those principles in much the same way as we have principles for the classical designs? Can we think of a process for doing this in practice? How do you actually do this? Describe that so that not everybody is reinventing a process for doing that. And can we actually do this for other types of research as well, not just experiments? And there's maybe a bit of an agenda for our project. All right, so here's the last slide. So what I've been talking about is relearning research methods. What we thought we knew as researchers is, yes, it's relevant, but it's relevant to a different problem than the problem we have when we're working with FRNs. So who is it that needs to research things, to relearn things? Well, researchers need to. The researchers working with FRNs need to often look at things in a different way. Students do, and the student supervisors do. And the student supervisors I pay particular attention to, because I've realized that those habits, if you like, research habits, students start to learn them when they're doing their training, when they're doing their masters, or if they go on to do a PhD, when they do their PhD. And those sort of set standards for how they'll do things later. So to me, it's most important that students are are helped to understand research and research methods in this way that is relevant to farmers and FRNs as they're doing their student research. And that is definitely not what is commonly happening at the moment. And what is it then that needs to be relearned? We need to be thinking about new sets of principles, and certainly not rules like the rule of you need 4 replicates, which is commonly quoted. We need adaptability rather than saying this is how you do it. These are the standards for doing a particular type of research, and we need to take into account multiple perspectives, not only the perspectives of the science establishment. For example, the rules in your faculty about how an experiment should be done if it's going to qualify for a master's degree, that's no longer adequate. All right, there. Sorry, I just want to say that last thing. That short article I wrote it for something, and then we didn't use it. That's available, and maybe we'll send it to you. We'll put it on our resources site later. But if you want a copy of that, it's only a couple of pages, and it basically says the same thing that I put in this seminar.